Good morning, dear professors and experts. Welcome to this webinar. It's a pleasure to have you here. My name is Yuyao Cheng, and you can call me Aris. I'm a product manager from the product department at BGI Tech. Thank you for joining us today, despite your busy schedules. Today, I'm going to introduce you to CycloneSEQ, BGI's Nanopore long-read sequencing platform, and explore how it's opening new possibilities to multi-omics research. Over the next 30 minutes, I will take you through 4 key areas. First, the rapidly evolving landscape of long-read sequencing and why it matters for your research. Second, the technical foundation and unique advantages of the CycloneSEQ platform. Third, real-world applications across multiple omics layers with concentrated data from actual projects. And finally, how BGI Tech's global infrastructure can support your research end-to-end. But before we get into the specifics, let me set the stage with a simple question. Why do we care so much about read length? Let's start with a brief history of sequencing technology. Look at this timeline. The Human Genome Project launched in 1990, and the draft was published in 2001, but with 8% of the genome remaining as gaps. These gaps consisted mainly of highly repetitive sequences, centromeres and telomeres, that short-read technology simply couldn't spy. It wasn't until 2022, enabled by long-read breakthroughs, that we finally filled these blanks. And the complete Y chromosome followed in 2023, and the pan-genome map in 1 year later. Long-read sequencing is driving genomics into the area of complete genomes. As researchers in the life sciences, we all know that every innovation in sequencing technology has fundamentally changed how we study biology. From the launch to the completion of the first truly complete human genome, it took us 33 years to finish reading this book of life. And throughout this journey, long-read sequencing has become increasingly critical. And the evolution of plant and animal genome assembly products has been linked the advanced in sequencing technologies. During the era dominated sold by Sanger sequencing and short-read sequencing, researchers could only produce draft genome assemblies for relatively simple genome. The subsequent adoption of high-C chromosome conformation capture technology enabled chromosome-level enables and representing a significant leap forward. With the advance of long-read sequencing technologies, assembly quality has progressed from high quality to ultra high quality standards, achieving telomere to telomere genome, which is T2T genome completeness and haplotype resolved assemblies. So why are long reads so important? Let me use a simple analogy. The short-read sequencing is like shredding a book into tiny pieces, each with only a few words, and then trying to resemble it. When you encounter the repetitive content, like gene, you have no idea how to put it back together. But with the long-read sequencing, by contrast, it's like reading entire paragraphs or even the full pages at once, directly spanning repetitive regions and clarifying the structural relationships. This capability enables breakthroughs in 4 key areas. First, the structural variant detection. We know that the structural variants, deletions, insertions, inversions, translocations larger than 50 base pairs, and the traditional short-read technology struggle with them, but with the long reads can span entire SV events, precisely determine the breakpoints and the variant types. As a recent Cell commentary noted, we are approaching the point where one assay can comprehensively detect all forms of genetic variations for the first time in history. And the second, the metagenomics. In complex metabiomes, short reads fragment genome beyond recognitions, and the long reads enable the complete metagenomic assembling genomes, and letting us recover high-quality genomes from organisms that constitute less than 1% of the community, and this changes our ability to study low abundance but the high important species. And the third, the transcriptomics. The central dogma tells us DNA makes RNA and makes protein, but the reality is far more complex. A single gene can produce dozens of different mRNA isoforms through alternative splicing. Short reads capture fragments, but the long read captures the entire transcripts from 5-cap to poly-A tail. Currently identifying the alternative isoforms and the fusion genes and the long non-coding RNAs. And as Nature Reviews Genetics recently called this a new area for the transcriptomes. And the fourth, it is the unique nanopore technology, epigenetics. Traditional methods require the harsh chemical treatment to detect DNA methylation, but with the nanopore sequencing, it can detect natively and in real time from the same data used for base calling with no extra steps. So with this context established, the question becomes, how do we make this technology accessible, scalable, and reliable for routine research? Now, let me introduce BGI Cyclone Nanopore sequencing platform in detail. We have developed two instruments for different throughput needs. One is the G400ER. It features an ultra-dense flow cell with over 30,000 active nanopores, designed for high output projects for large-scale projects where data volume is critical. And the other one is G100ER, and the G100ER offers dual flow cell of flexibility and suitable for small to medium throughput needs. But both two platforms support 5-MC methylation sequencing, and it can achieve 98% single molecular accuracy and reach the 99.995% synthesis accuracy at 50-fold coverage. And let me briefly explain how nanopore sequencing works to help you understand why we can achieve such long reads. The yellow components are motor proteins that control the uniform passage of DNA through the pore, and the blue components are the nanopore proteins responsible for detecting and distinguishing different bases. The membranes separate reaction chambers to generate electrical currents. During the sequencing, a single strand of DNA is guided by the motor proteins through the nanopore, and the different bases cause the same change in the electrical current. These electrical signals are detected and analyzed in real time and yielding accurate DNA sequencing information. And importantly, the DNA molecule remains intact throughput, no fragmentations or synthesis required. In theory, it allows unlimited read length, but in practice, read length depends on DNA integrated and the library preparation quality. And in our sequencing platform, the Cyclone sequencing data routinely produce reads as N50 over 20 gigabyte, and with the maximum length exceeding 100 KB. CycloneSEQ features complete independent intellectual property rights, and all three core components are developed in-house, and one is the key proteins mined from the deep-sea metagenomic database and engineered for robust stability, powerful capture, and high speed. And the other one is the sequencing flow cell, high density array design, low noise, and durable chemistry. And the last one is the base calling algorithm, and optimized using deep learning advances from the ASR field with the large-scale distributed training on massive databases. And the result is a platform that combines long reads, direct methylation detection, and the competitive accuracy with the operational advantages of a few controlled simply chain. But the significance on slides only tell part of the story. The real proof is in the data. So let's move to the applications. This is the core of today's presentation, and I will walk you through the real world cases of CycloneSEQ applications across different search areas. CycloneSEQ applications span 6 major directions: human resequencing, genome and the plant genomes, and the microbial genomes, and for the single cell or the bulk RNA sequencing and the epigenetics, I want to see not just what CycloneSEQ can do in theory, but what is already achieved in practice. First, for the human whole genome resequencing, we've developed a hybrid strategy for the maximized information which minimize the cost. Here it is. We combine five-fold coverage from the CycloneSEQ and with the 30-fold from our DNB-seq short read platform. This short reads can provide accuracy, SNP in detection, and excellent coverage uniformity. And the long read can provide the super indel calling and the comprehensive SV detection. With the SNP and indel performance equal to 30-fold pure only the long read sequencing, and the SV detection comparable to the standalone long read platforms, but at significantly reduced cost. Additionally, we can provide 5MC methylation information to database from one sequencing run. And when you have a large population cohorts, this cost efficiency difference can mean the difference between a study with hundreds of samples and one with thousands. The scale matters for statistically power, and its approach makes scale accessibly. And the professional supports we can provide the experienced pre-sales and after-sales teams, and we have the rich experience, a proven track record in large-scale cohort projects worldwide. Next, let's look at a key application of long-read sequencing, a landmark population cohort study on structural variations in 1019 samples was published in Nature on 2025. This study used ONT long-read sequencing at 16.9-fold coverage to analyze 26 populations from the 1,000 genome projects. And it generated a comprehensive structural variation map, and strictly 50.9% of insertions and 19.5% of deletions were not impressively reported in the database. And this clearly demonstrated the unique and value of long-read sequencing in high precision SV detection and analysis. And now let's look at a completely different scale, the bacterial genome. We all know the bacterial genome are small and typically a few mailing bases pair, but completeness matters enormously for the downstream analysis. With our CycloneSEQ bacterial de novo total solution, we can have the high-quality assembled genomes with zero gap, one contact, and complete coverage of bacterial chromosomal and sequencing encompass of all genetic information to prevent the misinterpretation and the gene functions caused by the missing regions. And we can robust resemble of complex regions with the long-read sequencing. We can have the repetitive elements and the GC extreme regions, and we can eliminating the assembly gaps and misassembly encountering to the short read methods. And we have the fast delivery, and you can have the optional 5MC and 6MA methylation information available. And for the data performance, the unique cellular assembly of 1 GB CycloneSEQ plus 1 GB DNA data yield a complete 3.9 GB cellular genome. And the cellular map compares 3 genetic rings and the inner to outer, the GC school, the GC content, and the annotated or featured genetics. For another bacteria, we use 3 GB of CycloneSEQ data with read length over more than 100 KB, and the N50 is more than 20 KB for assembly, and the subsequent polishing and resulting 8 contacts with a total length exceeding 6 megabytes, and 1.15 and 15 megabase secular chromosome 6 characters and one implicit likely due to the sample extraction loss. And this implication is clear. When other users are stating the environmental isolation of the industrial strains, you cannot obtain the reference quality genome routines and with no exceptionally. And for the plant and animal genome sequencing solution, in the assembly of genome and the plant genomes based on the Cyclone platform, the following genome assemblies have been completed. One is a finless purpose genome assembly. Use only 60-fold CycloneSEQ data, no PacBio or no ONT or other sequencing data. We achieve the assembly matrix statistically in this from a T2T reference genome, and the complete boost code genes is 95.5%. And compared with other T2T genome, which is 97.7% in the reference, and the single copy completeness is 96%, and with just 1% fragmented genes and only 1.5% missing genes. And the other one is the long stemate rice. It is a T2T assembly case. We use the CycloneSEQ and the HiFi and the HiC and the DNB6 sequencing. We can get a genome with 26 megabase contig N50 and 12 gap-free chromosomes and the clip telomere and the centimeters annotation. And the QV score is 52 and indicating the extremely high base accuracy and with the 98.6% boost code completeness. This isn't just an assembly. It is a foundation for understanding gene evolution, domestication, and the breeding potential in one of the world's most competent crop. And finally, one is the breeded dragon, a repetitive genome with 1.8 gigabytes. With the CycloneSEQ with HiC and the chromatin conformation data and the DNB6 data, the contig N50 reached 202.5 megabase and is exponentially high community with 6 completely gap-free chromosomes and 31 telomeres and 15 centimeters identified. The 200 megabase N50, to put this in principle, this is a newly chromosome level community from long read alone. And for the evolutionary biology, the developmentary studies for comparative genomes, this level of genome can changes what questions you may ask. And for the researchers conducting resequencing studies, assuming a high-quality reference genome is available, we recommend performing 50-fold sequencing on at least 10 repetitive individuals at last 2 groups of 5 individuals each to comprehensively detect SVs, indels, CNVs, and SVs. This provides the statistical power required for population genomics while ensuring the capture of a complete structure variation profile. We also offer promotional activities to support your research projects. Please feel free to contact our sales team if you are interested. And for the full-length transcript sequencing, the central challenge in transcriptions is that the gene expression is not just about the abundance, but it is the diversities. CycloneSEQ captures full-length transcripts from the 5 cap to the poly tail. We can see the complete isoform structure, and this matters for identifying novel isoforms that may have different functional domains or the subcellular localizations, and detecting fusion transcripts in center critical for diagnositions and the targeted therapy, and characterizing long-read sequencing RNAs that regular gene expression, understanding the expression by phasing transcript to the haplotypes. After introducing the application areas, we will now present the strategies for different research objectives. And we offer 3 sequencing strategies based on the biological question. You can use more than 20 gigabytes for the complex samples and the rare isoforms. And if you want you are interested in the tumor or the disease studies, you can use 10 to 20 gigabytes. And if you are just to do the plant and animal transcriptome characterizations, you can only use 5 to 10 gigabytes. And all the delivering more than 80% transcript or the gene detections. And the segregations plots confirm these data volumes efficiently reach the saturation and making your platform available for the isoform discovery and the quantifications. A critical question when adopting any new plant is, will my data be comparable to existing data sets? To compare the data analysis result with other platforms, we use UHR samples for library construction, sequencing, and the quantification coherence analysis. The left heat map shows high correlations for gene quantification, while the right heat map says the strong consistency at the isoform level. Based on the internal test data, these results available that our bulk full-length transcript sequencing solution delivers accurate, comparable results for both gene and isoform analysis. This means you can integrate CycloneSEQ sequencing data with published responsibility and the historical experience without any bias. And meanwhile, we are offering the trio credits and for your new CycloneSEQ platform full-length transcriptome sequencing to all customers. And if you have any sequencing needs, please feel free to contact us. And finally, let me briefly describe how BGI Tech supports research globally. BGI Tech's capabilities span the entire spectrum of genomics and the multi-omics sciences. Our 9 core platforms range from foundationally gene synthesis and DNA sequencing and to the advanced spatial omics, single-cell research, proteomics, and metabolomics. This comprehensive portfolio enables researchers to move simply from the genotype to phenotype and from the sequencing to the function. Whether your work requires short-read sequencing or the long-read sequencing or other spatial resolutions at the cellular scale, our integrated platforms can provide the technical foundation for discovery, and our core offering centers on the CycloneSEQ platform, which combine DNB and short-read technology with CycloneSEQ long-read sequencing to deliver comprehensive full-length solutions. And our technology infrastructure supports a truly global research network. We currently serve over 90,000 clients across more than 100 countries and regions, collaborating with over 7,000 partner institutions worldwide and participating in more than 30 national genome projects and with more than 4,800 high-quality publications. And we can proceed more than 700,000 samples for 1 year, and we have already pursued over 130 petabytes of genomic data and representing one of the largest responsibility of the life science information globally. And we provide a truly one-step solution for the sample preparation for diverse input types, including low input and the challenging samples, and for the library construction, optimized for your specific applications, and the sequencing across our full platform portfolio, and the DNB-seq for short reads, CycloneSEQ for long read, and the partnerships for complementary technology. For the bioinformatics part, we can do the standard pipelines or the other advanced or the customized analysis. And we have the comprehensive QC at every stage, because data quality is determined at the sample preparation, not just the sequencing stage. And at here, I want to highlight our BGI Poland laboratory, which is established in June 2022 with 14 staff members and capability for over 59,000 samples per year, and it provides localized services for European clients with comprehensive experimental production and quality control capabilities. And whether you need single technology or the multi-omics integration, we can provide customized solutions. Let me quick summarize today's key points. Today, we have seen what long-read sequencing is no longer a specific tool for the genome centers. It has becoming a standard requirement for the comprehensive biological discovery. And the BGI CycloneSEQ can provide this capability with fully independent technology and achieving an excellent balance between accuracy, read length, and the cost. From the bacteria to the plants, animals, or human, from the transcriptomes to the epigenetics, the CycloneSEQ has proven its value across the multiple domains. And BGI Tech's global infrastructure can mean you can access this technology with the spot, scale, and the quality control that major research projects demand. And if you are interested in the CycloneSEQ platform or our services, please contact us. Thank you for your attention.